Okay, good afternoon, everyone. Um, I want to start a class, and it is on southern blotting, northern blotting, and western blotting. I'm sure you all know about these techniques, and you all must have used them learned as a part of your genetic engineering, at least the biotech people. The why I'm dedicating a whole class just for this is because I want to take it to a level of analysis, especially those who are trying to write for examination, write CSAR or GATE. One will need to have analytical skills, and in today's class, using the familiar techniques of southern blotting, northern blotting, and western blotting. I'm sure everybody learned western blotting as a part of molecular biology lab, at least. Southern blotting, I think we have discussed ourselves in this course, and previously also you must have learned it somewhere. But even then, I'm still going to repeat. But then the goal is to uh, take ourselves to analytical level. Okay. So this is going to be an important class, and I would want that you all pay attention. You can ask questions, and then we will try to see what all we can do. Okay. So I hope you know what is blotting, right? If there is a spillage of water on a surface, somewhere something has spilled, we try to use a blanket or a paper, or in labs, if you know, there is something called as blotting paper. We use that paper to uh, pick up all the spilled water. It is like you're taking things up from the bottom. Okay, I mean you're cleaning up a surface, or you're actually picking this onto some other water or so. I mean paper or so. Somewhere I'm having a little bit difficulty with my computer. I think CPU is completely engaged. So 1 second, just be online. I'll check out if there is any other issue I need to check. No. Yeah, I found. Just give me a minute. Okay. Yes, sorry about that. So I hope I gave you a small introduction or told you what is blotting. In all the techniques that we are talking about, we have a gel usually, and the gel has some parts of some biocomponent like proteins or DNA, as in the case of electrophoresis, say inside. These are bands, and you're using a filter to pick up all these bands onto the filter. That is basically the blotting technique. That forms what is meant by blotting. Okay. So to start with, we have discussed this before about southern blotting. It is by the name of the scientist who invented this technique. And the goal or objective of this technique is to find or to know if a particular sequence is present in a given DNA sample or not. So it is all about hybridization. That is basically dependent on base pairing, right? I think in the last class somewhere we were also discussing about stringency of washing. Keep that in mind again too. So the technique goes simple. You isolate the genomic DNA, then we digest it. There is a restriction digestion step involved, and then you load the samples onto gel, electrophoresis gel. You run the gel electrophoresis, and the bands will partition themselves, or actually based on their size, because the mobility is inversely proportional to the size. Smaller the size, faster the mobility, or higher the mobility. After that, you are actually doing the blotting technique in this step, where you put a nitrocellulose membrane, and you allow the buffer from here to travel upwards because you're putting sponge, paper, towels, and so on. So while the buffer and other things move upwards in this direction, because nitrocellulose membrane has higher affinity for DNA, DNA gets stuck onto the nitrocellulose membrane. So now we have a membrane here with the bands, but you cannot see if there is a band, if there any of the DNA is not visible to us, just as such. So we need to now find out if there is the gene of interest or not. For that, the important thing is about how we use the probe. Our design of the probe in the previous one, where we were trying to decipher genomic imprinting, I did discuss about what is a probe and how it is radio labeled too. So the same thing is used here. I mean, it is basically the same technique in that place and here too. So we are using a probe that has sequence complementarity to the to the gene that we are trying to find if it is present or not. So wherever the seq band is present, the probe also goes and binds there, something like this. And then we wash away the excess probe, and then we expose it to X-ray film. And based on the radiation, there are usually the bands show up something like this here. Okay, dark bands usually are what we see in electrophoresis, upon hybridization and autoradiography. So before I go into northern blotting or anything, what I want you to also understand or try to have some insights into how we would analyze things, right? So I have assumed that there is a DNA molecule, or I'll try to reduce this. There is another one there. Okay, 1 second. Just give me a minute so that I can. So what I'm trying to now do is I'll give you a sample and assume that this is the genome of a bacterial cell. Okay, and we have a gene of interest. This is the gene I'm looking for, and I want to know if it is present or not in the sequence. And I have gone through all these steps: restriction, digestion, electrophoresis, and probe hybridization, as well as autoradiography. And say, for example, B1 implies that I have taken sequence from one particular bacterium. That is, I'm saying B1. It gives a band. What does that mean? You are all supposed to answer so that it will be better, more interactive. This is supposed to be much more interactive than normal. So if a band is visible, what does that say? It says that the gene we are looking for is present, right? Because the probe that we are having is complementary to the sequence that we are looking at. So what you can infer from any result like this is you can say that sample B1 has the sequence or gene that we are looking for, say gene X. Okay. And in the same plot when you did, and B did not give any band, what does that mean? It could mean that B does not have gene X. Okay, assume B does not have a band there. So it would mean that bacterial sample B does not have gene X. Now I have a situation where there is a different situation like this. I have something like this. What would that mean then? What does that mean? So probe, you have used the same probe. In one, it has given one band. In the B2, there is no band at all. In B3, you have 2 different bands. That could imply, try to follow the words that I'm using. I'm not, sometimes I will confirm that this is what it is. Sometimes I might say it could be. That means there are other possibilities too. Okay. So in B3, the most likely thing is that there are 2 copies of gene X. That is a possibility. Duplication can happen. Okay. Or if assume that when I take the probe, I have taken the whole length of gene X as a probe. Okay. I'm testing the whole length of it. Then it would probably mean that there are two copies of gene X in bacterial sample 3. The question again arises: why should one be in the size comparable to that of normal sample, and why should one have a smaller fragment? Why is that likely happening? In southern blotting, we are doing restriction digestion, right? Like you might be, I hope you're able to visualize, see that. So when you cut it with restriction digestion, say I'm trying to now draw for the sample B3. What may happen is that, say, this is the whole of gene X. This fragment, which I'm trying to indicate in a different color, probably I'll try. So this fragment from here to here is a larger fragment compared to from here to here. So one can give a bigger fragment corresponding to a larger size, and one can give a fragment corresponding to a smaller size. Now I'll have a little more complication to this so that it is about brainstorming. It's about thinking what are all the possibilities and then arriving at the most probable answer. Okay. So if we can't imagine, if we limit our imagination to what we want to see, that is wishful thinking, then we are likely to make mistakes. Instead, the best thing is to try to imagine all kinds of possibilities and then arrive at an answer. I will give a different situation. Say, for example, you have a situation like this, and you have another band like this. What would that mean? It could mean, okay, I don't have to speak about this band because it's normal. Say B1 is normal, so it is something like that. Why is this band larger? And why is this band smaller in B3? There are other possibilities too. Say, for example, I'm using a, if there is a gene, and I will try to use red, and this is normal normal gene X. There is a possibility that you may have several possibilities. In one case, what may happen is that you may have an insertion. In some other case, you may have a deletion. So in this case, you have there is a insertion into it. Because the majority of the sequence is still complementary to the probe that we are using, it will still give a band. And in this case, the same can happen because there is still some complementarity with that of the gene X, the probe that we are using for gene X, it might still give a band. It all again depends on the stringency of washing. Okay, if we increase the stringency, probably they might not bind. But because if you assume, as I said before, that the probe that we are using is a gene whole of gene length. I mean, the whole gene length is a probe, which could be at least hundreds of bases. That means there are more number of hydrogen bonds. That means stringency should be very high. That's sometimes not practical. Okay, so we have to keep in mind that when people work in the lab, they do a lot more times and optimize these techniques for each of their probe and samples, and only then they will conclude what their results are. But otherwise, I think I gave you ways of thinking. One of it, if there are two bands, there are two possibilities, especially if it is in bacteria, that means there are two copies. If you have, there are also other possibilities that within the gene there could be an insertion or a deletion. And there is also another possibility that I want you to remember. Say, for example, if this band didn't exist, what is it more likely that? Is it more likely that it is a deletion, or is it more likely that this fragment is smaller, like I have shown here? What could be? If both the bands are there, then I could say that there are two copies of gene X, whereas if there is only one band, that means I would suspect that there has been a gene deletion because it is bacteria. I think when we compare with the later one that we're going to discuss, you will probably understand it better. Now I'm changing it to the sample. In the last class, I told you, I think, no, not in your last class. Anytime we are trying to analyze an experimental data, you should know about the organism used. You should know about the biomolecule being analyzed, whether it is a protein, DNA, RNA, and so on. One should be aware of the principle of the technique. What kind of technique is used? In that, you would also need technical details, such as in this case, for example, the probe, and so on. A labeling technique, and so many other things. And then you should be able to interpret, right? So in the experiment that we have done just now, or we tried to analyze, I established that the organism is bacteria. You cannot apply the same rules if it were an animal sample, because there is a difference in the molecular biology of bacteria. There are a lot of similarities, but there are also a lot of differences too. If we didn't keep that in mind, we are likely to make mistakes. So we should also know whether it is DNA, RNA, or protein, like we are going to discuss today, and the technique and method and so on. Okay. So to consolidate what we have tried to understand until now about the skills in interpretation, we'll try to do some other one in this case. So here the sample is assumed from an animal. What is one of the difference between the DNA or the genome of animals and bacteria? Animals are diploid, right? We know that there are two sets in all of the animals, except in the germ cells, maybe, or the gametes. So there is a state of diploidy, isn't it? That means each gene has two copies. So now I'm trying to directly go through the same thing. Assume that there is a DNA isolated from an animal, and you have tried to study, say, gene X, or for difference, we will change it as gene Y. Okay, I'll just change into gene Y. Now, if the band is something like this, what would that mean? Assume that A1 is normal. We have already done similar experiments before, analysis. Try to recollect that. If there is one band and A1, I am saying, is normal, okay, what would that mean? That it must be homozygous. Say, because it's diploid, I need to tell you about if there are any other alleles or not. So whatever it is, A1 is homozygous. It has almost same level, similar sequences, okay, same size at least. One thing you have to remember is Southern blot cannot tell you exactly if there are any single nucleotide polymorphisms. It cannot distinguish between small changes. It cannot distinguish between substitutions or small insertions or small deletions. It can tell you the number of copies. It can tell you if there are any large scale differences. That it can, Southern blot can do. And now, if I had a situation like this from sample A2, if I had this, what would that mean? One is coming from maternal allele and the other one is from paternal allele. Both typically should show normal size if this is what is a normal one. Both bands should be there itself. What you can tell from this is that for sure A2 is heterozygous because it has two different alleles of a gene. It's not the same allele. They have two different alleles of a gene. And what is likely, what must have happened to it? What can you say about these two alleles? One is likely a normal one, similar to that of A1, but the other one seems to be mutated. And that mutation is most likely deletion because it's running much faster. Okay. Again, I'm not confirming that it is so. Okay, but I'm saying that it is most likely. That is what is most probable from these. And if I had a situation like A3, I'm going to add more complication to it. So I'm going to have something like this. So what can you tell about A3? A3 does not have a normal allele at all. This is missing. It has two, it is heterozygous, and it has two abnormal alleles. One of it is similar to that of A2, which is probably a deletion. And the other one is probably, it probably has an insertion. Okay, so you can think of, there are three different alleles that I'm talking about. One is Y, the other one is Y1, and the other one, take it as Y2, something like that. So now what can we tell about A1, A2, and A3? A1, I already established that it is normal. And now A2, the statement should come something like this. A2 is heterozygous, that is confirmed. It is likely to have one normal allele as shown here. And the second allele probably has a deletion mutation. So while A1 is something like Y, it is heterozygous for YY. A2 is, A1 is homozygous for YY. A2 is heterozygous and it must be having one Y and a Y2. This is one. Okay. What is the genotype of A3 in terms of gene Y? Then it doesn't have any Y at all. One copy of gene Y1 and one of Y2, something like that. I hope you're following this. It will, if you understood this, we will be able to solve this successfully, solve the problem at the end of the chapter well. So I'll move on to northern blot. Before that, I'll just summarize. In southern blotting, you can find out if there are multiple copies of a gene or not. First thing is you'll find out whether a copy of gene is present or not. Second thing is you might be able to figure out if there are multiple copies of a gene. Third thing is you can also try to find out if there are large scale changes such as large scale insertions or deletions. Okay, that is, those are the things that southern blot can tell you. Now we are looking at northern blotting. The objective of northern blotting is to find out if a gene, if a RNA component is present or not. It is about checking RNA. RNA W, say for example, we are looking at a specific RNA species W, and if it is present in total RNA or not. That's the objective of the experiment. Okay. So in bacteria, so you take the RNA sample, you'll run the blotting technique, everything is almost same. You'll use the probe also, which is very much similar, and then you will develop the technique. The only difference being is that, sorry, um, this is not, 1 second. Okay, I might have misplaced a picture, okay? I'm sorry about that. But you have done, you would have taken RNA sample, and the majority of the technique, rest of the technique remains almost same. So I'm taking about now, assume that the sample is from bacteria. And I'm looking at bacteria 1, bacteria 2, and bacteria 3. Bacteria 1 has a band, and bacteria 2 doesn't have a band. What can we say about bacteria 1 and bacteria 2? I'm going to make a statement. I want you to argue with me. Bacteria 1 has gene, assume that we are looking at W. Bacteria 1 has gene W, but bacteria 2 does not have gene W. Is it true or false? I'll repeat the question, repeat the statement. Bacterial sample B1 has gene W. I'm talking about gene. And B2 does not have gene W. What can you tell about it looking at this one? In one case, you see a very nice band. In another case, you do not see a band. So what would that mean? We are analyzing RNA, and there is a band. So this RNA can only come from gene W, right? So my first part of the statement is B1 has gene W. If I said that is correct, because if RNA is present, it must have come from the gene itself. It cannot come by itself. But the second statement is troublesome. If I do not have RNA, does it mean that gene W is not present? That one, nobody can say that. What if gene W is present, but it's not transcribed? Isn't it? So we have to be kind of philosophical in the clarity with which we are speaking about the communication of the results or even the interpretation. So if a band is there, it means gene is also there. If a band is not there, it doesn't mean gene is not there. Okay, it only means that RNA is not present. Why is that RNA present? There are two possibilities. One is that gene W is not there at all, or the second part is gene W is not transcribed. Okay, so keep those things in mind. Lack of a RNA in northern blotting, lack of a band doesn't mean lack of a gene. It only means lack of the RNA W, which could mean because of several reasons. If we have in B3, for example, I have more. Okay, what would that mean? Compare this the sample or the band from B1 and with that of band from B3. You'll find that you notice that the band 3 in B3, it is much more intense, which could be of two reasons. One of it could be how one performs the experiment. If you added more sample here compared to B1, okay, then you will obviously, you might get a darker band. But assume typically in every experiment, I'm just going to just put it like this. Say you used 100 micrograms of RNA. Okay, after processing everything, when you have taken for electrophoresis or when you isolate it, you took 100 100 micrograms of RNA, say for example, just for the sake of understanding. Then you have taken equal amount of RNA for all the samples. Okay, I'm telling you about the second possibility. So assume that equal sample is added, yet you're finding that one of the B3 has more or higher intensity of this band. What would that say? That would mean that you have in B3 it is less, in B1 it is optimally expressed, but in B3 you have more transcription of B3. It could be because of many other reasons. Okay, growth conditions. There could be a mutation, and something like that. So, what does Northern blotting tell you? Northern blotting tells you if a RNA species is present or not in the sample. It cannot definitively tell anything about the gene, whether the presence or not. But if the RNA is present, then the gene must have been present. Only then you will get the RNA band. But if the RNA is not present, it doesn't mean gene W is not there. It is one of the possibilities that this bacterial sample does not have gene W, or it might mean that that particular species is not, that particular gene is not transcribed in that organism or sample. And again, it can also tell us if the quantification of quantity of expression, how much of RNA, if you're going able to compare across these different samples, you can say now that B3 has highest amount of highest expression of gene W compared to B1 and B4. B4 has the least expression. Okay. And again, typically in a lab, all people, a majority of the people will do these kinds of experiments at least thrice from scratch, from isolation of RNA or DNA samples, and then trying to do it. They should be able to reproduce these the results. Only then there is consistency, and then that is reliable. Otherwise, not. So if we have it in plants, then we will have the complication as we have seen before. But I'll just I will try to say the same. So I'll have P1, P2, P3, and P4. In plants, compared to that of bacteria, what you should remember is diploidy. Okay. So if I have a band like this, P1, we will consider it as normal. What would that mean? I said normal, so it is homozygous and so on. If it is having less, what could that mean? If they both are same alleles, then it would be equal expression as shown in this case. But in P2, you do not seem to have normal expression. There is some decrease in the expression. What could it be? It could usually be because one of the allele is functional, but the other allele is non-functional. So what could happen is that, just give me, I'll try to erase this and write. So assume that this is the one allele in, I will give a name. So gene Y, okay. And then here we have Y. This is Y, that is normal one. And this is Y1, okay. There is some difference in that. So assume that Y produces about 10 transcripts per minute, whereas Y1 has some mutation. Because of that mutation in the promoter region, there is a mutation in the promoter region, because of which there is it is not producing as much as it does. right? So it is assumed that it is producing only 2 copies or even 0 copies. Okay, so if an organism is YY, it may have about 20 copies per minute, because if it is homozygous for the normal allele. If an organism has a normal Y and it's heterozygous, one is Y and Y1, then it is only having 10 copies per minute. Its transcription is happening, because only from normal Y there is transcription, but Y1 there is no transcription. If some organism is homozygous for Y1, then it will have 0, and then there might not have been any band at all in that case. Okay, so there are also many other possibilities that one can deduce. Say, for example, I will put this, and I'm going to put a copy like this. What could that be? It must have been a different allele altogether, Y2. Y2 could have been a situation where there are several possibilities for this case. One of it is that usually after every gene you have a terminator. That is where the transcription stops. But if there is a deletion in the terminator, there is no terminator, then the transcript produced will be very long, right? That could result in a longer transcript like this. Alternately, there is also a possibility where within the gene there is a large-scale insertion, because of which the gene size has now increased. That could also result in longer size RNA species. The same can be said for a sample like this, where there could have been a deletion within the gene that could reduce the size of the transcript, which will give it as a smaller band. Okay, so again, I'll try to summarize from northern blotting. Northern blotting is about trying to find out if a particular RNA species is present in the cellular total RNA of a cell. If the presence of RNA confirms the presence of the gene, but the lack of RNA does not confirm the lack of the gene. That is one thing you have to keep in mind. We have to rely on other techniques such as southern blotting to do this kind of analysis, to determine what is the problem. And RNA blotting can also quantify, if you have taken equal amount of RNA sample from all the different test samples that you're doing, then you can quantify relative to the normal, to the standard, saying one organism has more expression compared to the standard or normal one, or less expression. You can say that. In some cases, like if one is working with plants or so, even in bacteria, the size of the fragment of the band, the migration of the band will also tell us if there are any mutations in it. But we will have to associate those things with other techniques to confirm if there is a mutation or not. So large-scale insertion and deletions can be determined using northern blotting also, but only if the gene is expressed. Say if there was a mutation in the promoter region like this, then we don't have RNA of it. In that case, we don't know if there are any further mutations within the gene or not. Okay, I hope you understood how to interpret northern blotting. The last one is the western blotting. I hope you remember that. Now the sample is changing. Until now, we dealt with nucleic acids, which depend on base pairing or hybridization between strands and the base pairing between bases for probe and the sample to determine. For protein determination, we have to rely on different kind of things. We use antibodies. So the technique, I'm not going into the fundamentals of it, but you have to know about this step. Why is blocking important? Blocking is there in other techniques also, yeah, but this is a good enough place where you would want to learn. I hope you also have seen electroblotting in our molecular biology lab. We have done it as a part of molecular biology lab. And these are primary antibodies, those that have the CDR regions, complementarity determining regions of the antibodies that can bind to the epitopes. They are usually the monoclonal antibodies that are used. And then there is secondary antibody, which is specific to the primary antibody, constant region of the primary antibody, which is usually conjugated with an enzyme. That enzyme can convert a colorless substrate to a color bearing a compound with a color, which can be read as a signal. And or it can also be done using radiation, but this is much easier with the using the enzyme. Okay, that you can find it anywhere. You can always go there. So what I want to focus on is about how we interpret it. So as usual, W1 is normal. It gives one band. Assume this is the band. And protein, as I must say, Western blotting is about finding whether the protein is present, the protein of interest is present in a total protein of the cell or not, or in a mixture of proteins. It cannot confirmedly say if there anything about the RNA or the gene. We are now analyzing proteins. So if a sample shows a band, it means it must have had the RNA. That means it must have had the gene for that particular protein. Okay, or I should actually write it in terms of these. Sorry. So if there is a protein, I will call it as protein P. We will say it as P, and you have, it must have come from the mRNA for P, and that must have come from gene P. Okay, if a band is present, it means gene P is there and is transcribed. And if W2, sample W2 does not have a band, you cannot say that the gene is not present. You cannot say that it is not transcribed. All you can say is that the protein P is not present there. And similarly, you may also be able to say sometimes, yeah, and there is also one more situation. Yeah, I need to discuss that a little bit. Say, for example, I have taken a protein and unfolded it into a linear structure like this. Assume that each of these is an amino acid. Okay. Of course, no protein exists like this. Usually, they're all folded up based on the properties of the amino acid side chains and the composition in general. So if I fold it and assume that the epitope, these last three amino acids are the ones where the epitope is there, or that is where the antibodies are actually going to bind. Yes. Okay. Assume this is a protein. Okay. And I have monoclonal antibodies that actually recognize these last three amino acids. Just a theoretical situation. Say, for example, there was a mutation here. Because of it is a nonsense mutation. That means upon transcription and translation, you will produce a protein will be produced of this much length until here. If I use the same monoclonal antibodies as shown here, they will not be able to recognize this anymore. Okay. Those are some of the things that you need to keep in mind. You have to think of different possibilities. At Western blotting level, there are multitude of possibilities. So lack of band doesn't confirm that it is not there. Of course, it all depends on how much knowledge you have about your experimental setup. What I'm here describing here is trying to give you all the different types of scenarios one must have to think of. So there is also a possibility that not only nonsense mutation. What if a frame shift mutation happened here in this location? What would that happen then? These parts, amino acids would have been changed. In that case, the antibody would not be able to recognize this particular protein, or it might have been a different type of amino acids here, because of which the antibody will not be able to bind. Then too, one may not have an antibody, a band there. And as usual, that is one thing you need to keep. As usual, if there is less expression or a lighter band, assuming that one has taken equal amount of protein sample when you're doing the analysis, and if you find a lighter band compared to a standard, then one can say that it has less translation. It has less quantity, which could be regulated at the level of translation, or it could be that there is less of mRNA, which was because of less transcription. Okay, but there all could be. We cannot say for sure what is the reason for that. And so in Western blotting, one has to keep these in mind, that we are only testing about the amount of protein present. And we should know where exactly, it is a good idea to have a knowledge about where the antibody binds. Especially if one is trying to look at mutations, then one has to do a lot more, one has to be a lot more broad in terms of thinking of possibilities. What are all the possibilities? Like such as nonsense mutation or frame shift mutation and so on. In animals too, you can have, this is assumed that W4, it is basically about diploiding. Okay, you have 2 copies, so assume that this is an allele giving 2 copies of this much, and if one is giving less, that means one of the allele might be non-functional altogether. And as usual with mRNA, if you have something like this, then there could be a truncation in the open reading frame. Only then you are having a smaller amount of smaller size protein. So if you have a situation like this, say for example, there is a protein and the antibody is recognizing an epitope somewhere around here. Okay, somewhere epitope is somewhere here. And if a nonsense mutation happened in this place, the protein that is mutated protein produced is usually smaller. It is truncated, so it would travel faster. Whereas you can also think of it like this. If I had this is where it should stop, but the stop codon, say for example, the TGA that should have been as changed in the mRNA has become a sense codon, say GGA. And now translation would happen until the next stop codon comes by. So one can expect a longer protein size in that case. Okay, so Western blotting cannot tell us if the transcription happened properly or not. It cannot tell us the nature of the mutation directly. I thought of completing it, but it took longer than that. But I was arriving at this particular question. If possible, I would like you to take a print screen of it. Some of you must be having this already. By the next class, I would want that you compare and try to discuss this. I will also try to discuss the different situations here. Southern blotting of same persons, okay? Person 1, person 2, person 3, person 4, and person 5. Sorry, my bad. So I'm describing the problem, so you can try to come up with your own answers. Maybe you should submit it as an assignment. So there are 5 people. The person 1 is normal, and 2, 3, 4, 5 have different levels of mutations or some kind of problems are there. In each of the cases, you have to compare something like southern blot of 2 with the northern blot of 2 and western blot of 2. And that should be in relation to the normal person. This is normal, right? I'll put it like this. N, 1 is N, N, N. So you're comparing in two ways. You're comparing between the different blots, southern blotting, northern blotting, western blotting. And then you're also comparing the person 2 with that of the standard in each of these cases, and trying to arrive at the best possible solutions. If we have practiced this, you are also more than welcome to interact and try to find out the answers for it, and we will discuss in the next class. But before you come for the next class, I would like that you practice this, try to think, try to arrive at an answer, so that we will be able to benefit much better. Okay. Well, I think it took longer. I'll stop here. Any questions meantime you can ask, so that I'll be able to answer a few.